Rob Knight. He's going to be talking about Human Microbiome Project to the EarthMicrobiome Project. Power of 10 scales, sounds like we're going to hear about. University of California at San Diego. Rob?
Great. Thanks, Bruce. And I'd like to thank the organizers for putting together such an amazing day of science and for inviting me to participate in it. And especially thank Nigel for badgering me over and over again to show up. He can be very persistent, as many of us know. So it was almost exactly a decade ago that I first encountered Carl Woese's work through Warren Tate's lectures at the University of Otago, and since then I've primarily known his work through his enthusiasts at various other locations. Graduate work with Laura Landweber, and then postdoctoral work with Mike Yarris, and of course collaborating closely with Nom Pace while I was at Boulder. So it's really a privilege to be here, especially given the amazing talks that we've had. So what I'm going to tell you about is some of our large-scale and expansive projects to map microbiomes across our bodies and across the planet. And this really ties into this theme that you've heard about, Carl's discoveries being among the major discoveries in biology. And I would argue that they're actually among the major discoveries in science, because over and over again, science has really led us to redefine our place in the universe. So, for example, the Copernican revolution made us realize that our planet was not the center of the universe. The Darwinian revolution made us realize that rather than being at the top of a great chain of being, we were merely one twig on a large tree of life, where all organisms had an equal amount of time to evolve. The dark matter revolution is making us realize that what we can see around us, the stars and galaxies, only make up 4% of the mass energy of the universe, and the rest of it is dark matter and dark energy, where we don't even know what it is. And now the microbial revolution is leading us to reconsider our place in the tree of life itself. And of course, as Nom told you, we're just on this one tiny twig of the tree, and essentially all of the rest of this is microbial. And we're even starting to reconsider our place in our own bodies, because these estimates come from Savage's 1977 paper. So there's obviously huge error bars on the cell counts, but a widely cited figure is that we have about 10 trillion human cells that make up our body and 100 trillion microbial cells. So by that count, we might only be 10% human. But a figure that's more solidly established is the human genome consists of only about 20,000 genes, depending on what exactly you count. But the size of the human gene catalog in the microbiome ranges from 2 to 20 million genes. So by that measure, we're less than 1% human. So in the Human Microbiome Project, we spent $173 million of NIH's money, largely on this healthy cohort study, where together with a consortium of about 400 researchers across the country and across the world, we sampled 250 healthy people at up to 18 sites in the body, up to 3 time points, and collected 4.5 trillion bases of DNA, so over 1,000 times the size of the human genome in terms of the raw number of nucleotides. This was a collection of shotgun metagenomics and 16S ribosomal RNA sequencing, both of which you've heard eloquently explained by Joe and by Norm, who really introduced those techniques. And it was amazing how rapidly the human microbiome permeated the popular media, because it was in June that both Science and Nature devoted covers to the human microbiome. A couple of months later, it was on the cover of Scientific American, and later that month, it was on the cover of The Economist. So this gives you a sense that not only the scientific community, but the community at large is getting fascinated by this vast microbial majority that inhabits our own bodies. So one of the initial results was very puzzling, and so what I'm showing you here on the top, so each of these little bars represents the microbial community of one person, one time point, at one site in the body. And on the top, I'm showing you taxa. And so even if we focus in on the stool, for example, there's tremendous diversity in the kinds of microbes that different people have. So some people are mostly firmicutes, some are mostly Bacteroidetes, and so on. And so you might think that this would correspond to a tremendous diversity in function, but at least if you look at healthy people, and Larry Smyre is going to tell you tomorrow about what happens if you look at unhealthy people. But at least if you're looking at healthy people, you see this remarkable consistency in function, where you have these completely different microbial community assemblages that give rise to very static functional profiles in terms of these broad categories like purine metabolism, ATP synthesis, the ribosome, and so forth. So this was initially very surprising, but then it's exactly what you would expect to see looking around you in environments of larger scales. Because, for example, if I show you two grasslands and two rainforests, they look superficially very similar, but I can guarantee that none of the species are the same between this grassland in Africa and this grassland in New Zealand. Probably none of the genera, and it's only up at the family level that you start to see overlap. So this theme of very different species assemblages giving rise to very consistent functions is something that we see in a whole range of ecosystems, and our challenge now is really to understand those ecosystems and their functional differences. So to understand who's doing what in these microbial environments, we're largely turning to DNA sequencing, again, as you've heard explained by Joe and by Nom. Now, as I mentioned, in the HMP data set, we collected about 4.5 trillion A's, T's, G's, and C's. And so the problem is that the data look like this, and this is the first file of the HMP data. There's another 17,000 files like this, and this is about the first 0.1% of that first file. So you can see that you really have your work cut out for you trying to figure out who lives where in the environment. So to a large extent, what my lab has been focusing on is algorithms that take all of this data and turn it into pictures that you can make some sense out of. And when we apply this to the HMP data set, so this is what's called a principal coordinates plot of unifrac distances, we get this. So maybe you're thinking, hey, wait, I don't really understand that either. So let me just explain what this is. So each point on this plot is all of the complexity of a microbial community as measured by its DNA, boiled down into just one coordinate. So for example, this oral biofilm, all of its complexity goes down into just that one point on the graph. And you can immediately see some patterns there where the mouth and the skin and the vagina and the gut are very different from one another. And what's remarkable about this is that these patterns automatically emerge from an algorithm that knows nothing about the people, nothing about the samples. All it knows is the similarities and differences in DNA in the different microbial samples. So what this implies is that different parts of the body, indeed different parts of your own body, are totally different from one another microbially. So if I highlight the mouth and the gut of one subject, you can see that they're in totally different parts of this plot. So they're different, so what does that really mean? Well, it's only when we integrate the data with things like the EarthMicrobiome Project that we really understand what it means, because what we can do is we can take these two communities and we can ask, where on the planet are there two communities that are just as far apart in terms of their microbial community dissimilarity as the two communities I'm showing you? And so if we compare your mouth, for example, to a coral reef, you have these complicated mineralized structures covered in complex biofilms that perhaps your dentist pesters you about and so on. The microbes in the mouth are as far away from the microbes in the gut as the microbes in this coral reef off the coast of Florida are from the microbes in this prairie in Kansas. So they're entirely non-overlapping microbial communities with essentially no similarity. And what this means is that a few feet along the length of your body can make just as much difference to microbial communities as thousands of miles across the Earth's surface. So this might lead you to wonder, how stable are these differences in the microbiome? And my partner Amanda, who I have to admit has put up with a lot in the name of the microbiome, and I address this question by sampling our own microbiomes every single day for 6 months. And so what I'm going to show you, basically the dark points on here are me, the light points are Amanda, and the time series, each frame in this animation, put together by Yoshiki Vasquez and Antonio Gonzalez, two very talented grad students in the lab. Each frame in this is 1 day in the life of our microbiomes. And what you can see immediately is even within a person, there's tremendous variability in the skin microbiome. The oral microbiome is remarkably stable. And then you can see that the skin microbiome is intermediate between the two. And you can also see that over the 6-month time period, we retain our separate microbial identities, which is pretty remarkable when you consider that we live together and have all kinds of opportunities to exchange microbes with each other, right? And so I'll just rotate that around for you so you can see how that separate identity is made. And in related work with Noah Ferrer's lab at the University of Colorado, we found that we could actually match up your fingertips to the keyboard that you type on, and the palm of your palm of your hand to the computer mouse you use, in some cases with over 95% accuracy. So this came out in PNAS back in 2010, but more importantly, it was on CSI Miami, so you really know it's true. Anyway, this might lead you to wonder, well, where do our microbes come from in the first place? And if you have dogs or kids, as I do, you probably have some dark suspicions about that, many of which are true, by the way. So just like I can match you to your computer equipment by your microbial DNA, I can also match you to your dog by the microbes you share. But in all seriousness, how we're born has a tremendous impact on our first microbes. So this is work we did with Maria Gloria Dominguez Bello, now at NYU. And so what we're looking at here is the microbiomes of mothers an hour before they gave birth, and then of their babies 20 minutes after birth. And if you look over here in pink, we're seeing swabs from all over the bodies, including the meconium, of the newborn babies who came out the regular way. And then in red, we have the vaginal samples from all of the mothers. Whereas over here, in light blue, we have all of the body habitats from all the babies delivered by C-section. And then in dark blue, what we have is all of the skin communities of all of the mothers, so this profoundly altered initial microbiome. And so what we're investigating at the moment is whether these profound initial microbial differences may explain some of the later differences in disease incidence between C-section and vaginal births, including somewhat higher rates of asthma, of atopic disease, of food allergies, and even of obesity, all of which have now been linked to the microbiome. You may be wondering what happens after you're born. So this is work we did with Ruth Lay, who's now at Cornell, who studied the gut microbiota of one infant, her own, over the first 2.5 years of life. You're probably wondering why 2.5 years. That's when her kid was toilet trained. And it's a lot easier to get the fecal samples out of diapers than it is to fish it out of the toilet afterwards. So now each frame in this is going to be 1 week, and the question is basically over these 2.5 years, how completely does the infant come to resemble the adult fecal state? Is it a smooth progression? Is it jerky? Are there patterns in how that trajectory evolves over time, and so forth? And again, this is projected into the data frame provided by the Human Microbiome Project. And so if I'll just start this going, what you can see is sometimes there's tremendous change 1 week to the next, whereas other times there's very little change. And remember that the main thing that matters on these plots is how far apart a given pair of points is. And so what this means is that your kid really does look like a person, does look like a different person 1 week to the next, at least in terms of his microbiome, because some of these changes are bigger than the difference between any two healthy adults in the HMP. So this is just fascinating. So he gets antibiotics for an ear infection. You see this dramatic regression of the microbiome, followed by a relatively rapid recovery in this case. So that went by pretty fast, so I'll just rewind it for you and play it for you again. What you can see is with oral amoxicillin, you see this tremendous regression of the microbiome, undoing months of normal microbial development, followed by a relatively rapid recovery. And then when he gets to 2.5 years, he's more or less in the healthy adult state. And so this raises a whole lot of questions about what happens when you apply antibiotics in early life. During all this chaos, is it just the case that nothing that you do matters? Or could you in fact have a profound and lasting effect on the microbiome? And Marty Blaser has shown both in animal models, experimentally and epidemiologically in humans, that early life antibiotics make you substantially more likely to become obese later in life, as well as other complications linked to the microbiome. So although antibiotics can be life-saving under the right circumstances, it's probably not a good idea to assume that there's no cost to applying them, and they should be reserved for the situations where you're really sure that you need to apply them. This effect of development is so strong that we can even do it cross-sectionally. So this is work we did with Jeff Gordon's lab, looking at kids from ages 0 to 18. What we're looking at is the approach to the average of the adults in each population. So basically the y-axis is difference from the average of the adults. So the higher you are on the y-axis, the more different you are. What you can see is that in the African population in red, in the South American population in green, and in the US population in blue, the approach to the adult state is very consistent and happens during the first 3 years of life. However, where you wind up microbially in these three populations is totally different. So what I'm showing you here is principal coordinates data of the principal coordinates views of the same data. And you can see the most important factor is age, from the youngest kids in red to the oldest adults, 83 years old here in blue. You can see that the populations are very different from each other though. And the US population is very different from those two non-Western populations. And given that we now know that the microbes that you have in your gut affect how you metabolize drugs, ranging from digoxin to acetaminophen to cyclophosphamide, this is especially terrifying in the context of drug trials, because even these large-scale projects like the HMP have only characterized the microbiomes of healthy Western adults. And as soon as you look at kids, and as soon as you look at non-Western populations, you see these completely different configurations of the microbiome that you might never have seen in your original population. So if you test your drug in one population and deploy in another, you might see very different pharmacokinetics and toxicity. So you might be wondering, does all this diversity in the microbiome I'm showing you really matter? And if we take the case of obesity, for example, today I can classify with about 90% accuracy whether you're lean or obese based solely on your gut microbiome. So on the one hand, this is kind of a cool technical trick. On the other hand, I bet you're thinking, well, I bet you're not going to commercialize that because there's much easier ways to tell if you're fat than sequencing your gut microbiota, right? And I bet you can tell which of these people is obese without doing any DNA sequencing at all. But it turns out that predictive task, lean or obese, can only be done with about 58% accuracy based on the human genome and based on SNP models trained on hundreds of thousands of people, whereas as I mentioned, we can do it with 90% accuracy based on your microbiome. And the really cool thing is we can then go on to establish causality by using gnotobiotic mice. So this is work we did in collaboration with Jeff Gordon, and at the gnotobiotic facility at WashU, they're growing mice with no microbes of their own. And so basically what we can do is we can take microbes from a fat person, put them in a mouse, and we get a fatter mouse than if the mouse instead gets microbes from a lean person. But the really cool part is we can actually design a microbial community that we can inoculate the mice with that prevent them from gaining that weight when they're exposed to the obesogenic community. And so by being able to culture many strains using improved methods, many strains out of the gut of a single person, we can transfer some of that phenotype to mice, and then we can systematically dissect that microbial community to try to figure out which of those microbes are important. So we're able to slim mice down using this technique. We haven't slimmed down any humans yet, but we finally have IRB approval to move into trials in humans in the next phase of this program project grant, which is very exciting.
You're going to have trouble, since most of these bacteria probably won't grow.
Yeah, so the question is what happens when most of them don't grow? So what we did in this project is, so the two things that are problematic are overgrowth and doing it under strict anaerobe conditions instead of microaerophilic conditions. And so when you combine those, you go from getting about 5% of microbes out of the mammalian gut to about 60 to 70%. And a couple of slides I deleted basically show that you get nearly perfect spearman rank correlation in the EC numbers and the culturable component of the community. And you can also confer the phenotype using just the culturable component of the community. And so another thing that's really exciting about that is that proves that it's the bacteria that are doing it, not a virus, not a microbial eukaryote, not a cytokine, not a metabolite, not any of the other things you transfer when you transfer stool. But thanks for asking that. So then the next issue is how do I translate these community patterns, which most of what I'm showing you has been based on, to molecular mechanisms. And so we've been collaborating very closely with Peter Dorrestein at UCSD, and what I'm going to show you here is mostly based on the skin, which is a much more accessible sampling site. And basically what Peter did is he and his wife Kathleen collected about 500 samples from all over their bodies. And Peter ran them through the mass spec. I did 16S sequencing to look at the bacterial component of the communities. For full disclosure, their original plan was to do 1,000 sites each, but they got up very early in the morning to start, and they were still going at about 8 p.m. It was clear they were going to be up all night if they completed the 1,000 sites, so they decided that a few hundred would probably be enough to see interesting patterns. And sure enough, that's what we see. So when we look at the mass spec data, some molecules are broadly distributed over the body, others are localized to the armpit or the groin or the toenails, for example. We can do the same kind of thing with the bacteria. So the Staphylococcus are basically where you'd expect in the moist areas like the feet and the nose, Propionibacteria mostly on the head and shoulders, Corynebacterium more broadly distributed over the body. So with these detailed spatial maps, what we can do is we can use the spatial co-localization of the microbes and the metabolites to start to make conjectures about which microbes are performing which functions. So, for example, if we take take oleic acid, which is found in a lot of face creams and things, so you can see there's a whole lot of it on the face and on the hands here. If you look at where the Propionibacterium is, the Propionibacterium is in the same place as this oxidized oleic acid signal. So basically the oleic acid oxidizes on the head where there's Propionibacterium and not on the hands where the Propionibacterium isn't. And then we see similar effects with palmitic acid getting broken down to monoolein and monopalmitin. So what we can do there is we can take that conjecture based on spatial co-localization and bring it into the lab and ask if we feed that microbe that precursor, does it actually work? And so, for example, Peter was able to show that Propionibacterium indeed has the lipase activity that we expect when we test it in vitro. And so what we're trying to set up now is essentially a high-throughput culturing system and chemical screening system where we should be able to test these kinds of hypotheses in very high throughput to get direct evidence about which microbes are performing which functions in different environments. So this takes us to the EarthMicrobiome Project, and so I co-founded the EarthMicrobiome Project with Jack Gilbert, who's now at the University of Chicago and Argonne National Labs, and Janet Jansen, who's now the director of Pacific Northwest National Labs, back in 2010. And what this is a crowdsourced project where essentially we're bringing this kind of technology to all kinds of people who have interesting samples but don't necessarily have the lab skills or the data analysis skills to be able to make sense of it. And so by this point we've run over 30,000 samples, primarily supported by the Templeton and Keck Foundations. And so what's cool about this is what we can do with the scale of data is we can start to create predictive models that let us link environmental parameters to how often we see each taxon in each environment. So, for example, Jack did this really nice work on the Western English Channel, where from just a handful of microbial communities, a couple of dozen directly measured, as well as surface measurements of CO2 flux in the Western English Channel, he's able to predict the taxonomic abundance over the whole year, over thousands of grid points, just from that handful of samples. And so the power of this kind of modelling approach to take a few samples and extrapolate it on much larger spatial scales, whether it's our planet or whether it's our bodies, is immense. So what we're starting to do is to take satellite image modelling data and then link that to microbial assemblage prediction to figure out who's there. And then link that to the predicting the turnover of different metabolites, and then feeding that back into the climate models to understand how the microbes are changing their own environment and how that in turn will change the microbes again. And so, for example, for CO2 flux, what we can do is ground truth it against surface ocean measurements from the UNESCO Atlas. We're also applying this in the Earth systems model where over the whole planet, from soil microbial data, what we can do is we can take predictions under different climate scenarios of things like moisture shown here, temperature and so forth, and then we can predict what bacteria are going to be there. So this is predicting alpha proteobacteria, for example. And then we can predict the abundance of all the different enzymes using a technique called pie crust that we developed with Rob Byko and Curtis Huttenhauer. And then we can ultimately use that to predict CO2 flux over the entire planet and feed that into climate models. And it turns out that incorporating the particular kinds of microbes and how they respond to different environmental drivers is very important for refining those models and could really change the way we think about land use policy and different carbon fluxes, especially things like the relative effects of lightning strike and the permafrost, which is increasing with increased cloud cover. So coming back to the human microbiome, one sub-project of the EarthMicrobiome Project is this thing called American Gut, where Jeff Leach and I, back over Thanksgiving of 2012, Thanksgiving being a time when a lot of Americans are thinking about their gut for some reason, launched this project where the idea was to exploit the continued decline in cost of these sequencing technologies to bring them to anyone who was interested in finding out what bacteria they were inhabited with. Because the technology's declined in cost by over 1 million-fold in the last 15 years. You probably wouldn't pay $99 million to understand what was in your gut, but you might pay $99. Of course, it turns out that not everyone wants to know. This is Daniel McDonald giving a tour of the BioFrontiers building where we started the project and explaining to middle schoolers that we're going to use robots and lasers to look at their poop. And you can see a tremendous amount of individual heterogeneity in response there too. But a lot of people have been fascinated, and at this point we've raised over $1 million, mostly in $99 increments from members of the public, shipped out over 9,000 kits, deposited data from over 5,000 samples, so any researcher, any educator, any student, anyone at a company can just go there and download with a fully free and open source pipeline a huge number of microbiome samples, including a whole lot from people who are also having their human genome sequenced through the Personal Genome Project. And so this is leading us to find all kinds of fascinating associations. So you might have expected that your microbes can read out how old you are or how fat you are, but you might not have expected that they're also associated with things like how many hours sleep you get at night, how much exercise you get, and in some data sets, even whether you say you do that exercise indoors or outdoors. But the most compelling case that we have of why you would want to place yourself on this microbial map is the case of Clostridium difficile. So C. diff infections kill 14,000 people in the US every year. It's one of the most common hospital-acquired infections, and it's a particularly nasty form of diarrhea that's, as I mentioned, often fatal. And if we look at the stool microbial communities from patients with C. diff, this is just to reorient you on the map. If I highlight the stool communities from C. diff patients, you can see they're in this completely altered microbial state that looks almost more like a skin or vaginal community. So this is with Alex Karutz and Mike Sadowski at the University of Minnesota. So what's going to happen is that 4 of these C. diff patients are going to get what's called a fecal transplant from this one donor. And so the question, kind of like the question with the infant time series I showed you, is how fast is the transition going to be? Are they going to come to resemble the donor? Will they resemble the donor specifically? Will they stay there? All that kind of thing. So I'm going to start this going, and each frame in this is going to be 1 day in the lives of these patients who are going to the bathroom several dozen times a day. And what you can see is essentially immediately, so a day or 2 after the transplant, they all move into the healthy state right around where the donor is. And then they stay there for the several months of follow-up. And all of their clinical symptoms vanish within that couple of days. So this is really remarkable because antibiotics are typically only 20 to 30% effective against C. diff, whereas stool transplant is 90 to 90% effective. And the last large-scale clinical trial, doing a direct comparison, had to be ended early because the fecal transplant was so much more effective than antibiotics. It was considered unethical to continue withholding fecal transplant from the people on the antibiotic arm of the trial. So the question facing us now is essentially for how many other diseases can we identify a microbial dysbiosis? And the diseases that have been linked to the gut microbiome now include things you'd expect like inflammatory bowel disease and irritable bowel syndrome, things that are maybe more surprising like cardiovascular disease, colon cancer, and even things like rheumatoid arthritis. And then if the mouse models hold up, and there's obvious limitations with the mouse models, but in mouse models the gut microbiome has been mechanistically linked to things like autism, depression, and multiple sclerosis. So the challenge facing us as a field is basically how to identify these different dysbioses and then how to guide this aberrant microbial community back to health, whether it's by fecal transplant or whether it's by other microbial therapies, including things like more precisely targeted antibiotics, phage therapy, prebiotics, symbiotics, a whole arsenal of other things in the probiotics, of course, and a whole arsenal of things in the microbiome toolkit. So what we really need to do is not just develop this kind of map, which we're pretty good at doing right now, but develop more of a microbial GPS that tells you where you want to go on the map, and step by step what you need to do to achieve it. And ideally, we need to develop the system and make it so easy to use that even our children can use it. So you can imagine a kind of smart toilet where as soon as you flush, it's going to do some microbial readout and then deliver it to your smartphone, which, let's face it, I bet you're using in there anyway, and puts you on the map relative to your previous samples, tells you if you're going in a good direction or a bad direction, and maybe gives you some specific suggestions about what you should be doing in terms of diet or lifestyle or drugs in order to get to where you want to be on that map. So this might sound kind of surprising, And in fact, Bill Sanborn, who's our chair of gastroenterology at UCSD, told Larry just a few years ago that this was a procedure that would never be done at UCSD. Yet here he is right now holding a whole bunch of medical grade stool from open biome, about to do a fecal transplant, which is now a technique that he uses relatively routinely. You might be thinking that this is all kind of gross, but it is the nature of biology to be gross, and what could be more disgusting than literally picking the scab off the diseased udder of a cow and cutting your skin open and sticking it under, right? But that's the basis for vaccination, which is the most effective medical advance that we know of, saving billions of lives. The other thing that often comes up is, isn't the microbiome amazingly complex and we'll never understand it? But if you take the milk from that cow and you run it through the mass spectrometer, you'll find hundreds of thousands of compounds, most of which we have no idea what they do. But it turns out that cow's milk is a pretty good source of iodine, except if you live in the mountains, where at least a century ago, a prevalent and incurable chronic disease was goiter, another one was cretinism, both of which we now know are caused by iodine deficiency, and which were completely baffling at the time. And so cheap, effective interventions like iodized salt have eliminated these chronic, incurable diseases. The question is, in the microbiome, even without understanding the full complexity, will we be able to find therapies and interventions that are that simple and that cheap and that effective on a population scale, rather than having to be tailored to each individual? I'm just going to leave you with one last more sobering thought, which is, do you know when defibrillation was invented? Most people guess that it was maybe 50 or 75 or 100 years ago. But in fact, in the 1700s, people were running around London with Leyden jars. Essentially, there was a society formed for the rescue of persons recently drowned, where they were taking recent drowning victims and people who had collapsed for no apparent reason, and delivering electric shocks across the heart. And of course, this was enormously effective in bringing many of them back. The issue was that shortly after 1818, the practice stopped and they switched to other activities. That, of course, was when Frankenstein was published. And it's kind of amazing that as a society, they gave up a technology that literally brought people back from the dead because they didn't understand it and they were scared of it. And in many ways, we have the science communication challenge with the microbiome, a responsibility to understand it and to explain it in a way that these life-saving therapies will be broadly adopted and not forgotten the way defibrillation was. And if you think of the 150 years of additional lives that could have been saved between when it was abandoned and when it was next adopted, that's really remarkable to consider. Essentially, I think the challenge facing us is to build on Carl Woese's legacy and learn to control these microbial communities for the benefit of humanity on scales ranging from our bodies to our planet. So with that, I'd like to thank the many collaborators involved in this work, most of whom I've mentioned during the talk, so I won't read this list out. The huge number of people in my lab, currently and formally, who have contributed to these projects and others. And finally, thanks for being here today and for your attention. I'd be delighted to answer some questions.
Very extraordinary success of fecal transplant has reached a barrier. The FDA has interdicted it.
Yes, that is correct. So the FDA will not let you do fecal transplant for anything other than C. diff infection where the patient has failed antibiotics in the United States. Fortunately, there are many other countries where you can still do the research. So I was just in China talking to collaborators there. There's a lot going on in Taiwan, in the Netherlands, and in other countries. So although the US is certainly lagging behind in that regard, in many ways, that's not a problem that's not a problem that can't be got around if you have a sufficiently international perspective. Correct. So from a research perspective, it's not so bad. From a patient perspective, well, it also involves hopping on an airplane, which many people have done, or the AGA hates it when I say this, but you can also, if you Google DIY fecal transplant, there are very detailed instructions and videos on how to do it. However, it definitely has a substantial chilling effect. And one thing that I think is going to be very important is re-regulating fecal transplant in a different way, because currently the fecal material is regulated as a biologic, so you have to produce it the way you would produce a monoclonal antibody, which is very hard to document, especially the reproducibility of the process. Regulating it like an organ, or there's several other options that make more sense, would be a huge deal both for researchers and for patients.
Other questions? All right, Rob, thank you very much.
Thanks again.